My name is Alicia Popick and my topic is cellular respiration, oxidative phosphorylation, and the electron transport chain. Oxidative phosphorylation is the process in the cell metabolism by which respiratory enzymes in the mitochondria synthesize ATP from ADP, an inorganic phosphate, during the oxidation of NADH by molecular oxygen. The electron transport chain is the stepwise transfer of electrons from one carrier molecule as a flavoprotein or a cytochrome to another along the respiratory chain and ultimately to oxygen during the aerobic production of ATP. Steps of oxidative phosphorylation. 1. A coenzyme takes a pair of hydrogen atoms from a substrate molecule. 2. NADH and FADH2 send hydrogen atoms to the coenzymes located in the inner mitochondrial membrane. 3. Coenzyme Q, ubiquinone, releases hydrogen ions and sends electrons to cytochrome B. 4. Electrons are passed through the electron transport system and begin to lose energy. 5. At the end of the ETS, an oxygen atom accepts the electrons and combines with hydrogen ions to form water. Oxidative phosphorylation is the creation of ATP located within the mitochondria in a reaction chain that requires coenzymes and consumes oxygen. Oxidative phosphorylation creates more than 90% of the ATP, formerly ADP, utilized by the cells in the body. The main reactions take place in the electron transport system. The electron transport chain, ETS, is a series of integral and peripheral proteins located within inner mitochondrial membrane. Water is used for the basis of this process. The cells are easily obtain these ingredients because hydrogen is found inside all organic molecules and oxygen is an atmospheric gas. Step 1 in this process is when a coenzyme strips a pair of hydrogen atoms from a substrate molecule. When in the mitochondria, NAD and FAD are reduced through the citric acid cycle, producing NADH and FADH2. While in step 2, the NADH and FADH2 transport hydrogen or H+ ion atoms to coenzymes attached in the inner mitochondrial membrane. The electrons carry the energy and protons are released before the electrons are brought to the ETS. The NADH path includes the coenzyme flavin mononucleotide, abbreviated as FMN. FADH2's path continues directly to coenzyme Q, or ubiquinone. FMN and coenzyme Q are then bound to the inner mitochondrial membrane. In step 3, the coenzyme Q releases some hydrogen ions and passes the electrons to the cytochrome B. Then in step 4, along the electron transport system, the electrons start to lose energy in a series of small steps. This sequence is cytochrome B to C to A3. Lastly, step 5 is the end of ETS. The hydrogen pumps move the hydrogen from the mitochondrial matrix into the space between the inner and outer mitochondrial membrane. Also, chemiomosis is kinetic energy by passing hydrogen. The ion channels and the following coupling factors create a protein complex named ATP synthase. 6 hydrogen ions are pumped across into the inner membrane space. However, the hydrogen ion channels located in the inner membrane stop H+ to diffuse into the matrix. An oxygen atom then accepts the electrons and pairs with hydrogen ions to create water. ATP synthase is produced in the intermitochondrial membrane, which is the main goal of cellular respiration.